This next case study is a UPC++ application developed through a collaboration between Steve Hoffmeyer at Lawrence Berkeley National Lab, who has led the implementation effort, and the scientific applications team led by researchers at the University of New Mexico and Arizona State University. This work was developed in response to the COVID-19 pandemic, and I think the speed of developing what is a fairly complex distributed memory application really exemplifies the productivity associated with using UPC++. First, a little bit of background here on the problem involved. As I'm sure you know, the immune response is fundamental to determining disease outcomes for patients. What you hear about mostly in the news is antibodies, and antibodies are a very important part of the immune system, but not the only part. Antibodies can only stop a virus that is outside of the host cells. Antibodies are ineffective once the virus has actually gotten inside the cell, which is what the virus needs to do in order to replicate. Inside the host cells, combating the virus is the job of the T cells, another part of the immune system. So understanding how the T cells detect and clear the virus is really critical to understanding how the disease is going to progress and whether it's going to be resolved in a particular patient. To investigate this, the SimCove application team built a 3D agent-based model in which the agents are viruses and T cells, and it's a detailed three-dimensional model of the structure of the lungs. This is a quick overview of what SimCove looks like in terms of the implementation and the goals of the project. The ultimate goal is to model the entire lung at the cellular level. This requires modeling a huge number of elements because there are about 100 billion epithelial cells lining the surfaces of the lung, and there are hundreds of millions of T cells that can respond to viral attacks. The lung itself has a very complicated branching or fractal structure, which you can see in the lower right image and is well known in terms of the structure of the lungs. What that means is that a traditional continuous style of simulation would have boundaries everywhere in the simulation. Instead, the team is using an agent-based simulation. The time resolution is in minutes, but the goal is to simulate an entire lung for 20 to 30 days so that you can really look at the progression of a patient over a period of time during the illness. SimCove is written in UPC++, and it uses a distributed 3D spatial grid. The particles, which are the agents in the simulation, such as the T cells, move over time, and the computation, once they've moved into their position, is localized on a given processor. The load balancing is very tricky because of the movement of the particles. There's a lot of activity near an infection if the infection just starts in a particular part of the lung and then spreads out. The infection locations can spread and move over time, and new areas can erupt, which greatly complicates the load balancing requirements. There is a heavy use of RPCs in this application. As an agent moves between domains, it's easy to accomplish that movement with RPC and then to have the other processor that receives that agent to pick it up and perform the computation on it. It was very easy to develop the first prototype of this, and it produces fairly good distributed memory performance that avoids a lot of synchronization using the same semantics of RPCs that we've already talked about, namely that when one of the RPCs is executing on the remote side, you don't have to worry about synchronizing with other threads within that particular rank. And there's extensive support then for asynchrony that improves the overlap of communication and computation. This is what some of the main components look like in the simulation. These are the different types of agents that you see on the left-hand side, such as viruses, the epithelial cells, then you see the T cells, and so on. Each of these has a state machine describing its behavior. For example, a healthy cell that is infected transitions into one that's incubating the virus, into expressing the virus, and then being triggered by a T cell to become apoptotic, which is a form of programmed cell death that prevents the cell from producing any more virus. Without reading through all of the details of this, you can see a complicated control pattern and how this computation behaves depending on whether there's a virus in that particular region of the lung and where the T cells are in terms of attacking the virus in the cells and so on. One of the other complications in this code is its dependence on observational data, which is being used in three different ways. First, to figure out the parameters of the model, such as the rate of viral production by the infected cells. When the virus replicates inside the host cells, it takes advantage of mechanisms in the cell to reproduce itself, and exactly how that works differs by disease. Second, in order to validate the model, simulations are run and the output compared against observations from actual COVID-19 infected patients. These can include viral load measurements and imaging data, such as the CT scan of an infected lung, shown here. Third, observational data can be used to seed the model. That is, what's a reasonable starting point? What is the spatial distribution of the infection? So you're also looking at these patients to try to determine what does a typical load look like as the disease is just starting to form in the body. This figure, taken from the paper cited at the bottom, shows the quality of results of the SimCOVE simulation code. As mentioned in the previous slide, observational data is used to determine model parameters. These results show how, when using just 4 such parameters, the SimCOVE predictions of viral load shown by the orange curves can match the data shown with the red dots, taken from 8 different patients. As the paper describes, the mathematical measures of goodness of fit for the SimCOVE simulation are similar to those of competing models, shown by the gray curves. Additionally, the SimCOVE prediction of an earlier rise and more prolonged period at the peak level of viral load is consistent with the observations that the virus is most contagious early in the infection, prior to the onset of symptoms, when, almost by definition, there is no observational data to fit. The competing models are based on ODEs, and although they can match temporal distributions of viral load, they are unable to capture spatial aspects of the disease. This last slide is going to show visualization from an early prototype of this application. This is just a 3-dimensional cube with one small portion of the lung in it. So this is not looking at the whole lung. You are seeing the virus spread, and in this case, you'll see it now sort of oscillate as the viral load and the T cells are working against each other, and then now getting to the edges of the simulation boundary. I think this is a very exciting use of UPC++ to solve a timely and critical problem and to really understand the immune response to COVID-19.